Today, I will be talking about separating DNA mixtures by computer to identify and convict a serial rapist. This is joint work by Cybergenetics and the Kern Regional Crime Laboratory in Bakersfield, California. In the summer of 2013, California residents were fearful in an East Bakersfield neighborhood. A man was brutally attacking and sexually assaulting women and children. He planned his rapes and took steps to avoid being caught. His first victim, a 17-year-old student, fought back and miraculously escaped, but 2 weeks later, he struck again. The second victim was a 22-year-old mother, asleep in her bed next to her baby. A masked man woke her with a glove over her mouth. He tied her up, raped her, and got away. Police later collected DNA evidence, a purse strap used to tie up the victim, a phone cord brought by the rapist, and the victim's cell phone hidden by the rapist in her couch. 2 weeks later, he struck again. Leaving for work before dawn, a mother was attacked by a masked man wearing gloves. He blindfolded her with duct tape and zip-tied her hands and feet. He bound her 3 young daughters. He raped the woman, molested her 11-year-old, and fled. Investigators took items from the crime scene and clothing from the hospital, including the victim's shirt and pants worn during the rape. 2 weeks later, a mother was asleep in bed with her 9-year-old daughter. Awakened by a sound, the woman saw a masked man wearing gloves in her bedroom. At gunpoint, he blindfolded them both with duct tape and bound their hands with zip ties. He raped and robbed the mother and washed her in a bathtub. But the daughter freed herself, reached a telephone, and dialed 911. This is a recording of the 9-year-old's 911 call.
911. Hi. Hello, 911.
Yes, this is Guy. He has my mom naked.
Okay.
And I hear he had me blindfolded.
Okay, how old are you?
9.
9. Where's your mom at now? What?
She's not my dad. She's not my dad.
The girl escaped to a neighbor's home. The rapist fled with $5,000, but in his haste, he dropped 2 zip ties in the road. County forensic scientists analyzed the evidence items in the 3 rape cases. They developed a major DNA profile from the zip ties found in the road and uploaded it to CODIS, the National DNA Database. On October 9, 2013, the crime lab learned that CODIS had matched the zip tie DNA to Billy Ray Johnson. By early October 2013, the Kern Regional Crime Laboratory had developed considerable biological evidence. They had 37 swabs and cuttings from 3 serial rape crime scenes. These included garments, blankets, apartment surfaces, skin, body cavities, telephones, and zip ties. But the data were low-level DNA mixtures containing 3 or 4 people that defied human interpretation. The crime lab had recently started using TrueAllele casework in their laboratory. TrueAllele is a computer system that automatically interprets DNA evidence. As shown on the left, a visual user interface permits entry of data and asking of questions and later review of results. Data is uploaded to a database server and interpreted and matched by machine using parallel computing. The crime lab had only 4 parallel processors at this time, whereas Cybergenetics had 40. So the lab sent the serial rape crime scene evidence to Cybergenetics. In early October, Cybergenetics received the evidence data from the Kern Regional Crime Lab. This is what the quantitative mixture data looks like for one of the biological evidence items as short tandem repeat data. On the x-axis, we see the size of DNA fragments with the allele number in increasing order of size indicated on top of the peaks, 9, 10, 11, and 12. And vertically, in relative fluorescence units, we see the relative amounts of these alleles. The data is from the zip tie in the road at locus D13. The jury icon on the top right of the slide indicates that this was one of the PowerPoint slides eventually shown at trial to the jury. The DNA data in this case was largely inconclusive to human review, unable to reveal minor contributors to the mixtures. This is because human review draws a threshold, shown here as a red line, through the data. Over the threshold, peaks are considered to be in, and their peak heights are ignored. Under the threshold, the peaks are discarded and the alleles vanish altogether. This throws out considerable identification information from the informative DNA data. The TrueAllele computer instead uses all of the peak height data without thresholds. It separates the mixture into genotypes by considering every possible genotype solution. As shown here, overlain on the data is a pattern made of colored rectangles. There's one person's allele pair shown in blue with two alleles at 11, explaining a tall peak. There's another person's allele pair shown in orange at alleles 9 and 10 as a second allele pair. And a third person's allele pair is shown in green as a 12-12 genotype. Adding together these different alleles produces lower peaks at 9 and 10, a tall peak at 11, and a very short peak at allele 12. The added together rectangles have heights that are very close to the peak heights of the underlying data, producing the same pattern. Since this pattern explanation very much explains the heights of the data peaks, it has a very high likelihood, it's a good explanation, and those genotypes end up with high probability. Poorer explanations move these rectangles around with different heights and at different allele locations and have lower probability. By considering every possible genotype solution and analyzing 100,000 different possibilities, the computer works out the probability of each of these genotype possibilities for the separated contributor genotypes without having to look at reference information from individuals, and it can use all of the quantitative data. Shown here is the major contributor at the D13 locus from the zip tie, separated out from the other two genotypes by computer. This genotype was objectively determined from the data without knowing anything about the comparison references. On the x-axis, we see 3 out of about 100 possible allele pair genotype values. 911 has a 19% probability, as indicated on the vertical y-axis of probability, numbers that add up to 1 or 100%. The 1011 possibility is 13%, and the 1111 genotype possibility has about 68% of the total probability. DNA match information tells us how much more an individual matches evidence than a random person. As shown in the display, we again have the evidence genotype as the blue bars, just as on the last slide, with probability indicated by the heights of these bars. But now we've added brown bars that show the chance of a coincidental genotype. Comparison can be made between the genotype that we obtained from the evidence as the pattern of blue bars, that's the genotype after having seen the data. We see it's very different from the brown bar pattern next to it at these allele pairs of coincidence, showing that the data provided information about this genotype from the evidence. Comparison to make a match statistic focuses our attention on the genotype of interest. Here we're going to be comparing with an 1111 genotype value. So we draw a red circle from that individual, here a suspect, on the right around what's happening at the 1111 allele pair. Here we see the 68% genotype probability is a tall blue bar, which we saw in the previous slide. But now that's compared to the shorter 7% probability in brown of a random person having an 1111 genotype. This ratio of the tall 68% probability at 1111, the suspect's genotype, after we've seen the data in blue, divided by the chance of a random person having this genotype by coincidence in brown of 7%, gives a ratio of 10. 68 divided by 7 is around 10, and this is the DNA match statistic. It's the probability of an evidence match divided by the probability of a coincidental match. A match statistic is computed in the same way for all 15 genetic loci. And we see the match statistics here on a plot where the names of the 15 loci are indicated on the vertical y-axis, and the extent of the match statistic, also known as a likelihood ratio, is shown on a scale from 0 to 500 on the horizontal x-axis. Because these genetic loci are biologically independent of each other, their match statistics are independent as well, and therefore we can multiply together the match statistics at all of the 15 loci, the lengths of the bars that we're seeing here on the plot, in order to obtain the total match information across all of the data for this separated genotype relative to the individual we're comparing with. We can now answer the question, to what extent is the suspect present in the evidence? Reading off the math formula for match statistics, in plain English we can say that a match between the zip tie and Billy Ray Johnson is 211 quintillion times more probable than a coincidental match. How big is 200 quintillion? Well, 100 quintillion is a 1 followed by 20 zeros, as written out here on the slide. That's the same as taking 10 and multiplying it times itself 20 times. 10 times 10 times 10 times 10, etc. Or 20 powers of 10. In the match statistics that we describe, we'll be talking about the powers of 10 because that's the natural way of talking about match information, and it makes it easier to compare large and small numbers. The TrueAllele processing had been done without having any reference data, solely on the evidence. In late October of 2013, Cybergenetics received the reference data from the Kern County Laboratory. Shown here is the table of match statistics for the first crime scene. There are 13 rows corresponding to the evidence items that were analyzed, and there are 4 columns on the right for the different comparisons that were made to 4 different individuals. The last column, shown in red, is Billy Ray Johnson. Looking at the first row, we see that the items swabbed from outside the kitchen window, having separated genotypes that match 2 of the people in the apartment, but not to Billy Ray Johnson. The 6 means a match statistic of a 1 followed by 6 zeros, or a million, and the 3 is a match statistic of a 1 followed by 3 zeros, or 1,000. Moving down to the third row with the purse strap, we see a separation of the mixture into at least 3 individuals, matching 2 of the victims at the apartment, as well as Billy Ray Johnson with a statistic of 2, or 100. The next row shows the phone cord, which the assailant had brought into the apartment, matching Billy Ray Johnson with a statistic of 5 zeros, or 100,000. Continuing down through all 13 items, we reach a phone which separates into different genotypes, 3 of which are matching the victim with a statistic of 19, that is a 1 followed by 19 zeros, or 10 billion billion. Another individual with 2 zeros, or 100, and also matching Billy Ray Johnson with a statistic of a 1 followed by 5 zeros, or 100,000. The second crime scene's match tables shows informative data producing match statistics from these mixtures for 11 out of the 12 items, and 3 matches to Billy Ray Johnson. At the third crime scene, TrueAllele produced informative DNA match statistics for all items except the cooler, separating the mixtures into their component genotypes and producing match statistics relative to the compared individuals. Here we see in the fourth row the bathtub handle produces 6 zeros or 1,000,000 to the victim and 3 zeros to Billy Ray Johnson. And towards the bottom, the zip tie left in the roadway contains the victim with a statistic of 100,000 or 5 zeros, and a much larger statistic for the major contributor to Billy Ray Johnson of 20 zeros. The informative DNA mixtures that match Billy Ray Johnson are shown here in this reduced composite table. It shows the separation and matching of 3 individuals from a complex mixture in the first row and the third row, as well as from the back of the shirt in the 6th row. Also, it shows how separating mixtures introduces highly probative evidence. If we look at the last row of the zip tie found in the roadway, Billy Ray Johnson had been found on that from a CODIS hit with a statistic of 20 zeros. However, he could have claimed that he just dropped his zip tie in the roadway. By finding the victim of the crime, shown to the left with a statistic of 4.5 in the tens of thousands, that establishes that the zip tie came into contact with the victim, for which there's no obvious explanation for a defendant, and a separated mixture produces that probative power. So these DNA match statistics tend to tell us what happened at a crime scene and corroborate victim statements. Cybergenetics produced a DNA match report which was presented to a Kern County grand jury in December of 2013. This led to the indictment and arrest of Billy Ray Johnson. At the trial in March of 2015, the jury heard how 8 different items across 3 crime scenes were connected to Billy Ray Johnson. They also heard the role that these different items had played in those crimes, connecting them to what had occurred. Validation studies help establish the reliability of a technology. There have been over 30 validation studies done for TrueAllele DNA mixture interpretation, 7 of which have been published in peer-reviewed journals. This paper, TrueAllele Genotype Identification on DNA Mixtures Containing Up to 5 Unknown Contributors, published in the Journal of Forensic Sciences, was co-authored by Cybergenetics and the Kern County Laboratory and had useful information that the jury heard. The paper gave match error rates showing the false positive rate in a specificity table, table number 8. Shown inside the blue square are the number of false inclusions grouped by match statistics. In particular, we see for 200 picograms and 3 or 4 contributors that there are 0 false inclusions in the 4th row, which over in the orange box corresponds to 3 powers of 10 or 1,000. The green box is showing that this is out of millions of comparisons. So what we can say is that based on this validation study, we have not seen any false inclusions in over 1 million comparisons when the match statistic is at least 1,000. The Kern County Crime Laboratory runs its own in-house TrueAllele computer system, and they continue to process the evidence samples in this case. The crime lab's independent TrueAllele processing produced a lab report with match results completely concordant with the cybergenetics match statistics. In April of 2015, the jury found Billy Ray Johnson guilty on 24 counts. The following month, the Eastside Rapist was sentenced to life in prison without chance of parole, plus 423 years. For more information about how TrueAllele objectively separates DNA mixtures and produces accurate DNA match statistics, please visit Cybergenetics' website, where there are courses, newsletters, a newsroom, many presentations, publications, and webinars, as well as access to a YouTube channel with dozens of presentations. Thank you very much.